Hello, everyone. Thank you for joining us for this tech talk. Today, we'll talk about proteomics profiling in tumor xenograft models. We will discuss the challenges in this field and the innovative strategies we can explore to overcome them. So now get started. Xenograft models plays a crucial role in oncology drug discovery. They offer a controlled environment to study human tumors, allowing researchers to evaluate treatment effectiveness and safety. Moreover, these models provided valuable insights into tumor biology and therapeutic responses. However, the presence of human mouse mixture in xenografts poses challenges for proteomics profiling. Distinguishing human and mouse proteins becomes difficult due to the high percentage of common peptides shared between those two species. Tumor purity refers to the proportion of human cancer cells within the mixture of mouse non-cancerous cells. Our previous study revealed that tumor purity in PDX models closely resembles patient's tumor, although it can vary depending on the type of cancer. These variations are influenced by difference in stroma content and immune infiltration, which in turn are affected by host mouse's immune systems. However, in traditional proteomics profiling for xenograft tumors, a common approach is the run and separate strategy, where human and mouse proteins are analyzed together using mass spectrometry. This method faces a significant challenge in accurately distinguishing the singles due to the substantial overlap of peptides between human and mouse. To address this issue, we conducted feather tests. In our study, we simulated PDX tumor samples using human and mouse cell lines at varying percentage of mouse cells. We employed the tandem mass tagging, aka TMT, for proteomics profiling. The results were intriguing. We found that many human proteins were falsely identified as differentially expressed. The heat map on the left side of the slide shows that the number of differentially expressed proteins and ensured depths between sample pairs, labeled with the percentage of human cells. Notably, the number of depths increased rapidly with a higher difference in mouse cell percentage between samples. The power curve demonstrated a superconductivity relationship between the number of depths and the mouse cell percentage. To understand the falsely identified depths, we investigated the relationship between depth category and the PSM ratio. Here, PSM stands for peptide spectrum match, and the PSM ratio represents the ratio of PSM counts from human unique peptides to the total PSM counts from both human unique peptides and human mouse common peptides. A higher PSM ratio indicates more quantification of a protein by human unique peptides. Our analysis involved the differentially expressed proteins analysis between mouse-contaminated samples and pure human samples. The results revealed an intriguing pattern. Upregulated proteins in mouse cell-contaminated samples tended to have lower PSM ratios, while downregulated proteins in mouse cell-contaminated samples had higher PSM ratios. We also observed that the fold change of the upregulated proteins was negatively correlated with tumor purity. This finding strongly suggests that many human mouse common peptides were misclassified as human peptide, leading to the false identification of depths. To explore the impact of mouse cells on proteomics profiling in real-world PDX models, we conducted proteomics analysis on 5 pairs of liver PDX models, each with varying percentage of mouse cells. In this study, the PDX samples labeled with the A suffix represented the original PDX samples, while those with the B suffix were the demoused samples processed using a mouse cell depletion kit. The results revealed a robust and positive correlation between the number of depths and the percentage of mouse cell present in the samples. These findings align remarkably with what we observed in the proteomics data from the cell line mixture. This investigation provides valuable insights into the influence of mouse cells on proteomics profiling in real-world PDX models, emphasizing the significance of addressing mouse cell contamination in such studies. The correlation observed between the number of depths and the mouse cell percentage underscores the need for cautious interpretation of proteomics data in PDX tumor research. Our differentially expressed protein analysis between paired Here's our PDX models considering both the original and demoused samples lead to significant discoveries. Without removing mouse stroma cells from PDX tumors, we found that approximately 30% to 40% of the differentially expressed proteins identified in pairwise comparison were false positives. Moreover, about 20% of those truly differentially expressed proteins could not be identified, regardless of the threshold used for calling differential expression. Additionally, as the percentage of mouse cells in the samples increased, the false positive rate escalated while the true positive rate decreased. Based on these results, we highly recommend adopting the separate xenograft strategy for more accurate proteomics profiling of xenografts. By removing mouse stroma cells before proteomics analysis, we can significantly reduce false positives and improve the identification of truly differentially expressed proteins. During our internal study, we compared paired PDXO-PDX phosphor proteomics data. PDXO is a PDX-derived alkaloid. The PDXO-based catalyst enrichment analysis demonstrated higher consistency with the drug's mechanism of action. One potential explanation for this finding is that the PDXO models lack mouse content, reducing the likelihood of proteomics profiling being influenced by mouse cell contaminants. To further explore this, we did a phosphor proteomics analysis to directly compare the original PDX samples with demoused PDX samples. I will talk more about this 4D phosphor proteomics approach later. To ensure a comprehensive analysis, we carefully selected 3 PDX models with varying mouse content. Our analysis focused on 2 main aspects. Firstly, we examined the consistency of phosphorylation sites proteins between the demoused and original samples. Secondly, we compared the abundance of these commonly phosphorylation sites proteins between the 2 sample types. By investigating these aspects, we aim to gain deeper insights into the impact of mouse cell contamination on phosphor proteomics data in PDX models. These findings will contribute to the development of more accurate and reliable methods for phosphoproteomics analysis in xenograft tumors, ultimately advancing our understanding of tumor biology and therapeutic responses. The Venn diagram we generated illustrates the consistency of phosphorylation sites proteins between the demoused PDX sample and the original PDX sample. Our analysis revealed that the percentage of both common phosphorylation sites and proteins between these 2 sample types were only slightly lower than compared to the percentage observed between technical replicates. Moreover, we made a noteworthy observation of a negative correlation between this percentage and the mouse cell percentage. In our study, we used label-free quantification, aka FQ, to measure the abundance of phosphorylation sites and proteins in both the original PDX models and the demoused PDX models. We then calculated the rooted mean square error, RMSE, and the Pearson's correlation coefficient to compare the FQ measurements between these groups. The analysis revealed that PDX models with higher mouse cell content shows less consistency in the abundance of phosphorylation sites and proteins compared to their demoused counterparts. Particularly, when the mouse cell content was relatively high, the accuracy of phosphorylation protein measurements was significantly compromised. For example, in model LY 2345, with the mouse cell content of 19.5%, the RMSE of a phosphorylation protein FQ was twice as high as the model PA 2425, with the mouse content of 4%. The striking difference emphasizes the critical importance of removing mouse cells to achieve accurate phosphorylation protein quantification. As shown by the scatter plot, we observed a strong negative correlation between accuracy measurements and mouse cell content. More specifically, as the percentage of mouse cells in the samples increased, we observed a noticeable deterioration in the RMSE, and the correlation between measurements also decreased. These findings underscore the necessity of minimizing mouse cell contamination in phosphoproteomics analysis to ensure reliable and meaningful results, providing a better understanding of tumor biology and potential therapeutic targets. To summarize, in the field of proteomics, addressing the challenge of common human mouse peptides in xenograft models is crucial to avoid inaccurate quantification of proteomics data. Our comparative analysis between the original model and the demoused model established a clear correlation between the number of depths and the level of mouse cell content, highlighting the significant impact of mouse cell contamination on proteomics data in xenograft models. To overcome this issue, we compared two strategies for proteomics profiling, the separate then run approach and the run then separate approach. The investigation showed that the separate then run strategy outperforms the run then separate approach in providing more reliable proteomics profiling of xenografts. In phosphoproteomics, the consistency of phosphorylation sites and protein between the original PDX models and the demoused PDX model is slightly compromised, particularly with high mouse content, about 20%. Mouse tissues significantly affect PTM quantification accuracy, warranting mouse cell depletion for PDX models with substantial mouse cell content. In conclusion, addressing mouse cell contamination in proteomics and phosphoproteomics analysis is crucial. By employing appropriate strategies and careful data interpretations, we can enhance data reliability, contributing to the advancement in cancer research and precision medicine. These insights will aid in developing targeted therapies and personalized treatment options for cancer patients. Now let's switch gear to talk about some other approaches we take at Crown Bioscience to advance proteomics profiling. One of the approaches we employ to advance proteomics profiling is 4D-TIA quantitative proteomics. This cutting-edge technology combines the power of ion mobility dimension with traditional LC-MS, resulting in remarkable advantage for proteomics analysis. The integration of ion mobility dimension provides enhanced separation, which significantly boosts sensitivity and specificity in our analysis. This proves especially valuable when dealing with complex samples and identifying low abundance proteins. In addition, the implementation of data-independent acquisition DIA offers its own set of advantages. DIA allows us to achieve more comprehensive coverage of our sample and greatly enhance the data reproducibility. Unlike data-dependent acquisition DDA, which selects specific precursor ions for fragmentation and analysis, DIA captures and analyzes all ions in the wide MORZ range. This feature ensures that no valuable information is overlooked and enables a more thorough and consistent analysis of the sample. The combination of the ion mobility dimension and the DIA in 4D-DIA quantitative proteomics opens up new possibilities for uncovering previously hidden insights in proteomics research. By harnessing the full potential of this approach, we can gain a deeper understanding of complex biological systems and make significant strides in the field of proteomics and cancer research. In one benchmark run, 4D-DIA can identify more than 7,500 proteins from a single injection of 200 nanograms of HeLa elixir, while conventional LC-MS proteomics analysis usually requires microgram level samples to detect about 5,000 proteins. To summarize, with this additional ion mobility separation of the peptides on top of the regular LC-MS methods, with DIA strategy, we'll be able to achieve higher sensitivity, deeper protein identification, better data reproducibility, and more reliable quantification. For post-translational modification analysis, we use the method called 4D phosphoproteomics. This technology combines additional ion mobility separation to overcome the challenge of isomerization, ensuring accurate and reliable identification of phosphorylation sites. The improved separation capability significantly enhances the depth of coverage for phosphorylation analysis. In conjunction with this approach, we utilize the immobilized metal affinity chromatography, aka IMAC, to selectively enrich phosphopeptides. This strategy, coupled with the use of targeted antibodies, effectively reduce sample complexity, allowing for more focused and sensitive detection of phosphorylation events. The results of this advanced approach have been remarkable. In various cell tissues, we have been able to identify over 10,000 phosphorylation sites with high confidence. This represents an impressive 50% increase in the number of identified phosphorylation sites compared to the traditional LC-MS method. By adopting this cutting-edge technology, we are paving the way for a deeper understanding of protein phosphorylation and its role in various cellular process, signaling pathways, and disease mechanism. Thank you all for joining us once again for this webinar. If you have any questions or need further information, please feel free to ask them now. If you think of any questions after this webinar, you can always reach out to us by email, and we will be glad to assist you. Thank you.
Thank you very much, Dr. Mao, for that presentation. We have received a few questions from the audience, and I will start with the first question right now. Will the additional step of using the mouse depletion kit affect the global phosphorylation status?
Yeah, we performed the correlation and PCA analysis on both the original PDX samples and the DMOS samples. The results showed that the global phosphor proteome remains consistent after the removal of mouse content using the mouse cell depletion kit. Therefore, the use of mouse cell depletion kit does not systematically alter the status of phosphorylated sites.
Thank you. Thank you very much. Next question. How do you determine the mouse cell content, and what cutoff do you recommend for considering the use of the separate then run protocol?
We have developed a highly sensitive and cost-effective NGS-based method to determine mouse content with sensitivity up to 0.1%. This method was published in Nucleic Acid Research in 2020. Generally, if the mouse content is higher than 20%, we strongly advise removing mouse cells before proteomics analysis. For mouse cell content is between 10% and 20%, it is also recommended to remove mouse cells before proceeding with further proteomics analysis. Thank you.
Thank you for the answer. And the next question that we have. Do all proteomics data at Crown Bioscience employ the separate then run protocol?
Yes. We have utilized the 4DDI platform to measure proteomics data for over 200 PDX models and several mouse genetic models. For all these models, we removed mouse cell content before conducting proteomics profiling. This approach ensures that the reliability and accuracy of proteomics data and helps us to deliver high quality and consistent results to our clients. Thanks.
Thank you very much for that answer. It looks like we have two questions remaining that we have time to ask. So the first question is going to be, could you provide more details about the traditional run and separate strategy?
Certainly. In the traditional run and separate strategies, researchers typically analyze PDX tumor samples containing a mixture of human cancer cells and mouse stromal cells. After data acquisition through mass spectrometry, computational algorithms are employed to assign peptides to either human or mouse proteins. Thank you.
Fantastic. Thank you very much for that answer. And the final question that we have here for the session is, what are the main methods used in the run and separate strategy?
In our study, we evaluated three types of run and separate protocols. The first one is the full peptide approach. This method involves searching both common and species unique peptides against the unified human and mouse protein database. The second approach is unique peptide approach. With this approach, common peptides are discarded to ensure an unambiguous peptide assignment. contributing to a more accurate identification of human and mouse proteins. The third one is the human or mouse-only approach. In this method, peptides are exclusively inserted against either the human or mouse protein database, focusing solely on identifying proteins from the respective species. Thank you.
Thank you very much for answering all the questions, Dr. Mao. This concludes the Tech Talk for today. Please feel free to try to send us a message or any comments directly, or visit the Contact Us page on crownbio.com. And we look forward to having you join us for another Tech Talk in the very near future. Goodbye.